Hi, I'm Jessica, back again for something totally different. So for the last 3 years, we've been running this Mixotrophs and Mixotrophy working group, and wanted to share some updates because this is our last year. Harkening back to Monday, when we were talking about activities here that have been started from, with a long lead time in the past. So this working group actually was originated from this 2016 summer workshop when Holly Moeller presented on mixotrophic modulators of the biological pump, as well in conjunction with the NSF and ASLO funded ecological dissertations in aquatic sciences a couple months later. We're led by a group of 5 of us, Nicole Millette at VIMS, Becky Gast at HUI, who's now at NSF, myself, Holly Moeller at UCS Santa Barbara, and Karen Stamieskin at Bigelow Lab. But most of this work is done by our working group members. Are Nicole and Emily in the audience? Can you guys raise your hands, wave? Okay. We have 2 members who are here in our meeting, but there was a group of about 20 plus of us that worked on all of this stuff. And we've been really busy. So we've had 2 hybrid and in-person meetings with another one coming up next week as a research incubator. We've had 10 webinars, bi-monthly webinars on different research topics, which are all available on YouTube, thanks to May, who's wonderful. And they range on topics from modeling and biogeochemistry to remote sensing. Maybe we can get mixotrophs from space, from PACE. I think I'm totally excited about it now. And just published like 3 days ago is our white paper from our working group on mixoplankton and mixotrophy future research priorities. And these we had 5 main research priorities that came through this working group. We got everyone together, everyone presented a couple slides on their top research priorities, and we tried to group these in major themes, and we all ranked them. We used Mentimeter. Our group likes Mentimeter. And we came up with these 5 main topics, which I'll kind of briefly talk about next. So first is evolution. And all of this is in our paper. So, you can also just tune out now and go check out our paper. We posed evolution because, phagomixotrophy has arisen in multiple evolutionary lineages. Actually all major eukaryotic lineages except for diatoms. They exist on this trophic continuum from basically photosynthesizers that eat to heterotrophs that are kleptoplastics that steal chloroplasts and continue to photosynthesize. So there's been there's been 2 key transitions. The first is the gain of photosynthesis. For example, in kleptoplasty and mesodynium, there's a range of kleptoplasty in different strains, as well as the loss of phagotrophy, so in diatoms. And studying evolution can be really important on informing common ecological selection pressures, as well as the impacts of future global change. And some of the questions that we posed we think are important in kind of like a 5 to 10 year time scale to answer, how does phylogenetic ancestry shape traits and function across mixoplankton, and how does ecosystem selection alter the expression of mixotrophic traits and function in diverse populations? So going from evolution, then looking at kind of the cellular function, we then posed that traits and trade-offs was our second research priority. So this idea of traits and trade-offs come from this sense of, that organisms contain different traits, and there's a trade-off between selection pressures for different organisms to be dominant in the ocean. So we're interested in quantifying these relative costs and benefits and looking at the mechanisms underlying mixotrophy trade-offs and how these traits and trade-offs differ between constitutive versus non-constitutive mixotrophs, which are really just the autotrophs that eat versus the heterotrophs that steal. And throughout this whole working group, a common theme is that we do know where mixoplankton live. So we know where there are plankton that can utilize the multiple trophic pathways, but we don't actually know where the biogeography of mixotrophic activity are. So there's, in our paper, we summarize the areas in which we think that mixotrophy may be advantageous in a changing ocean over autotrophy and heterotrophy. So you could, I won't walk through this entire figure, but the different mixotrophs that would be more advantageous in oligotrophic gyres versus coastal systems versus polar seas are distinct. And they vary from the heterotrophs that steal versus the autotrophs that eat. And that there's a number of emerging technologies such as transcriptomics, metagenomics that can reveal biogeographic patterns, but it really, like a key research priority is how metabolic profiles of these mixotrophs will really vary across lineages. So there's still a lot of work to be done, mostly on understanding what drives this activity and where it is. Another theme that was really obvious very early on is that there is a huge knowledge gap on mixotrophy, mixoplankton. I'm still struggling with this nomenclature. We adopted a different nomenclature. So mixoplankton and biogeochemical cycles. And some of the key questions that we're interested in are, how our cellular composition and extracellular biochemistry vary as these mixoplankton navigate the trophic landscape. How does micronutrient limitation work? Do they do autotrophs that eat more when they are micronutrient limited? And then associated with this, how are nutrients acquired from different trophic strategies allocated, and how that has implications up the food chain, so to trophic transfer efficiency and biogeochemistry. And summarizing this, are mixotrophs of superior prey quality. And then undergirding all of this is that we need better in situ methods for studying mixotrophs. This was kind of our little stab on R.V. Belafonte with the mixotroph focus on it, showing just the different ways that we can be studying and encouraging this research. So yeah, with that, check out our recent white paper. We have a methods paper focusing on these major themes and the methods that we can be using to answering these questions, going to be submitted later this year to Frontiers in Marine Science. And we're welcoming any new ideas for future iterations of this work. So thank you very much.